Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the Scientist.com and Axlead Drug Discovery Partners, Inc. webinar titled Improving Preclinical Decision-Making in Obesity and CKD with Translational in Vivo Phenotyping. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host. Today, we're fortunate to be joined by Kosuke Hitaka and Dr. Shuichi Takahara, who are research scientists with Axlead Drug Discovery Partners, Inc., who will discuss how in vivo phenotyping using patient-relevant models can improve preclinical decision-making. But just before we get started, a few notes on housekeeping to help you get the most out of the event. First, this webinar is being recorded and will be made available to watch on demand shortly after the live event. Next, if you take a look at the interactivity panel on the right-hand side of your screen, you'll see 3 tabs corresponding to chat, Q&A, and handouts. If you happen to experience any technical issues, please first try refreshing your browser, as this tends to fix most issues. But if that doesn't work, you can reach out to me using the private chat function, and we can help you to troubleshoot. We'd also love to hear from you, so please send us any questions, thoughts, or comments using the Q&A tab. And finally, you can also explore a few relevant resources in the Handouts tab to learn more about some of the content presented today. And with that, I'm very pleased to hand things over to the first speaker, Kosuke Hitaka. Hitaka-san, thanks so much for joining us, and the floor is yours whenever you're ready.
Good day, everyone. Thank you very much for joining this webinar. My name is Kosuke Hitaka from Axlead Drug Discovery Partners. Today, I will introduce how we support obesity drug discovery by improving translational understanding between preclinical and clinical studies. First, I would like to briefly introduce Axlead. Axlead is a Japan-based integrated drug discovery solution provider, founded as a spin-out from Takeda and Teijin Pharma. We support global clients by leveraging farm-originated research platforms, extensive drug discovery data, and experienced scientists. Today, Axlead has around 400 employees, including approximately 200 scientists with advanced degrees. We also have more than 1,000 legacy projects and over 1.2 million available compounds. Axlead provides end-to-end solutions across the drug discovery value chain. Our capability includes screening, chemistry, DMPK, safety, assessment, and pharmacology. We also cover omics, bioinformatics, pathology, and animal models. We support multiple disease areas, including oncology, immunology, CNS, cardiovascular, and kidney disease, metabolic disease, and musculoskeletal disease. By integrating these functions, platform technologies, and disease experience, we help clients accelerate drug discovery research and make better preclinical decisions. This is today's agenda. Today, I will first describe the key challenge in obesity drug discovery. Then, I will introduce our solutions focused on differentiating efficacy beyond weight loss and predicting safety-related liabilities. Finally, I will summarize how our capability can support next-generation anti-obesity drug development. Obesity is a major driver of systemic disease, including diabetes, cardiovascular disease, and stroke. As shown here, many anti-obesity drugs have been developed over the years. However, most of them failed due to safety concerns or insufficient efficacy. Recently, GLP receptor agonists have marked breakthrough in obesity pharmacotherapy. GLP-1 receptor agonists show strong efficacy in terms of weight loss. However, several key limitations remain, including weight regain after treatment discontinuation, mass reduction, and adverse effects such as emesis and nausea. These limitations highlight the need for next-generation therapies with improved profiles. Two major strategies are currently being explored. First, novel MOA anti-obesity drugs, which aim to provide differentiation from GLP-based therapies. Second, dual or triple therapies based on GLP-1, which aim to enhance efficacy and address these limitations. For these next generation therapies, two key requirements are critical. Efficacy differentiation from GLP-1 therapies and addressing GLP-1 limitations such as nausea and muscle loss. To successfully develop these therapies, it is essential to generate data supporting these aspects, both clinical and preclinical setting. This is essential to establish clear clinical value beyond existing GLP-1 therapies. However, there are significant challenges in evaluating these aspects in preclinical studies. First, differentiation based solely on weight loss is limited. Body weight reduction alone doesn't provide sufficient information to distinguish new therapies from GLP-1 therapies. Second, predicting clinical reliability is difficult. For example, rodents don't exhibit vomiting, making it difficult to evaluate emesis risk. In addition, muscle mass is typically assessed using terminal and low sensitivity methods. These limitations make it difficult to generate translationally relevant data and to capture clinical important endpoints. To address these challenges, we provide translational solutions based on advanced phenotyping platforms. Our approach focuses on comprehensive phenotyping beyond weight loss, translational surrogate assay for emesis, and non-invasive and high-sensitivity assessment of muscle mass. Now, I would like to start with the first challenge, differentiating efficacy beyond weight loss. First, I will show how we characterize the composition of weight loss. Using echo-MRI and CT-based analysis, we can quantify fat mass versus lean mass, as well as regional fat distribution, including visceral and subcutaneous fat. Importantly, these measurements are performed non-invasively and longitudinally. This allows us to determine whether weight loss is driven by fat reduction or lean mass loss and where the fat reduction is occurring. In this way, our platform enables us to evaluate the quality of weight loss, which is critical for differentiating new therapies from GLP-1 therapies. We also perform mechanistic profiling of anti-obesity therapies. For example, we evaluate central appetite regulation using CFOS, energy expenditure, gastrointestinal function, and lipid absorption. We enable us repeat mechanism-based differentiation beyond weight loss and support functional differentiation from GLP-1 therapies. Now, I would like to move the second key challenge, addressing GLP-1 limitation, particularly emesis-related side effect and muscle loss. One major limitation in preclinical study is the lack of emesis in rodents. To address this, we use multiple translational surrogate assays. We combine established assays such as conditioned tasty aversion, pica behavior, and gastric emptying with proprietary salivary-based assay for emesis prediction. By integrating these assays, we can robustly predict emesis risks. Another critical point is muscle mass loss. Traditional evaluation rely on terminal measurements, which are invasive and lack sensitivity. Our platform enables non-invasive and longitudinal quantification of lean mass. In addition, we evaluate the functional impact of muscle loss by measuring energy expenditure. This allows us to link muscle mass to metabolic outcome and better assess its critical relevance. In summary, next-generation obesity drug development will require differentiation beyond weight loss and prediction of safety liabilities. We provide translational platforms that enable detailed phenotyping, mechanistic understanding, and improved prediction of clinical outcomes. In addition, we have extensive experience supporting drug discovery projects across diverse anti-obesity mechanisms. Based on these capabilities, we are well positioned to strongly support your next generation anti-obesity drug discovery programs. We would be happy to discuss your project in more detail. Please feel free to reach out if you are interested. Thank you very much. If you have any questions, please feel free to ask.
Fantastic. Thank you so much, Hitaka-san, for a great presentation. And with that, I'm pleased to welcome the next speaker, Dr. Shuichi Takagahara. Takagahara-san, thanks so much for joining us today, and feel free to get started whenever you're ready.
Hi, this is Shuichi Takagahara from Translational Research Business Unit at Accelerate Drug Discovery Partners. Today, I'm going to talk about improving preclinical decision-making in CKD with translational in vivo phenotyping. This is today's agenda. I'll briefly review global burden and unmet needs in chronic kidney disease, CKD. Then I will discuss key challenges in translational CKD research. Then I will present our solutions for addressing these challenges, including translational animal models and MOA analysis platform. Finally, I'd like to summarize today's presentation. This slide shows global burden and unmet needs in CKD. CKD affects approximately 850 million people worldwide and is defined as abnormalities of kidney structure or function that persist at least 3 months, typically assessed by a decline in glomerular filtration rate, GFR, or markers such as albuminuria. Despite this substantial burden, therapeutic options still remain limited. Current therapies can slow disease progression, but there is no curative treatment available. As a result, there still remains a significant unmet need. However, drug discovery in CKD is quite challenging. Here, I will outline key challenges in translational CKD research. The International Society of Nephrology provides guidance for preclinical studies in translation that includes selecting the right model that reflects human disease and conduct studies by using robust study design and evaluating disease models by assessing target engagement. However, there are two key challenges. First, a lack of clinically relevant models. Many animal models show albuminuria with only limited GFR decline, as well as high variability and inconsistent disease progression, and poor representation of multifactorial human disease. Second, a lack of mechanistic validation that is characterized by complex, compartmentalized kidney structure, diverse cellular responses, and limited ability to link observed effects to on-target effects. To address these challenges, we have developed an approach. Let me share our solutions for addressing the challenges in CKD research. To address a lack of clinically relevant models, we have established translational models with robust, reproducible phenotypes, including AXI mouse and SHE rat. To tackle a lack of mechanistic validation, we have built an integrated platform for MOA analysis, including these technologies and human samples. By combining better models with deeper mechanistic analysis, we aim to improve translatability to humans. I will explain each topic in more detail in the following slides. This slide shows our highly translational CKD models, AXI mouse and SHE rat. Among vascular damaged or glomerular injured or tubulointestinal injured CKD models, compared with these conventional CKD models, AXI mouse and SHE rat show robust, chronic, and reproducible albuminuria together with progressive GFR decline. That is the key hallmarks for CKD. In addition, these two models show a lower variation in disease onset and severity, which makes it easier to detect drug effects. I will explain each models in the following slides. Let me start with AXI mouse, which is genetically defined with collagen 4A5 mutation derived from a patient with Alport syndrome, which leads to marked loss of collagen 4A5 expression, particularly in glomeruli. They develop albuminuria at 7 weeks of age, followed by elevated blood urea nitrogen, decreased GFR, and eventually end-stage renal disease, recapitulating Alport syndrome, and progression to CKD. Pathologically, these mice show abnormalities in glomeruli, interstitial fibrosis, and inflammatory self-infiltration. Regarding body weight, they develop comparative to wild-type mice until at 17 weeks of age. Regarding kidney function, they show progressive albuminuria and a decline in GFR, which leads to a reduction in survival at 25 weeks of age. Next, we evaluated effects of irbesartan, which is clinically used for CKD, a class of angiotensin II receptor blocker. Irbesartan was dosed from 7 weeks of age to 20 weeks of age. Irbesartan treatment significantly reduced albuminuria in AXI mice, consistent with clinical responses in CKD patients, as well as kidney injury marker NGAL and inflammatory marker TNF-alpha. These results indicate that AXI mice are translationally relevant model for evaluating therapeutic effects in CKD. We are also evaluating another clinically relevant drug, sparsentan, which is a dual inhibitor of endothelin receptor and angiotensin receptor, but is approved for glomerular diseases. Next, I will introduce SH-RAT as multifactorial CKD model. SH-RAT spontaneously develop hypercholesterolemia and hypertension, which leads to histological changes of focal segmental glomerulosclerosis, FSGS-like phenotypes, including segmental sclerosis, mesangial growth, or vacuolation of foot cells. They develop albuminuria at 7 weeks of age, followed by elevated BUN, decline in GFR, and kidney death, similar to CKD patients. Next, we evaluated effects of irbesartan in SH-RATs. Irbesartan was dosed from 10 weeks of age to 24 weeks of age. As a result, irbesartan significantly decreased albuminuria and showed a toward tendency of improvement of GFR decline, as well as plasma creatinine levels. These results indicate that SH-RATs are also translationally relevant models for CKD. Next, let me introduce our integrated platform for linking phenotype to mechanism, which enables mechanistic understanding across kidney compartments for clinical translation. From organ-level search or organ-level analysis to molecular and single-cell analysis, we can connect phenotypic changes with underlying mechanism. In addition, we can also provide human samples, including urine, blood, and kidney tissues that can be used for early POC obtaining. Let me summarize today's presentation. Translational CKD research is challenging due to lack of clinically relevant models and mechanistic validation. To address these challenges, we can provide translational animal models with robust reproducible phenotypes, and our integrated MOA platform enables mechanistic understanding across kidney compartments for clinical translation. As a next step, we would be happy to start a conversation about your project. You can share your question, and we can start a discussion. Based on that, we we can propose a study plan. Please feel free to contact us if you are interested in CKD efficacy studies. 1 more minute, please. I'd like to introduce a booster. A booster is designed to accelerate candidate generation by starting from validated science, rather than beginning from a blank state. It leverages more than 1,000 pharma-originated legacy projects and pre-qualified compounds and data. This allows clients to jumpstart at the lead generation or lead optimization stage, rather than starting from hit identification. By combining these assets with AI-powered design, make, test, and analyze cycles, a booster aims to deliver promising leads for approximately 6 months and support candidate nomination for about 1 year. Another important advantage is IP ownership. Clients can gain full ownership of co-created IP, while also benefiting from a shorter lead time and integrated scientific support. Thank you for your attention. I'd be happy to take your questions.
Fantastic. Really nice job on the presentation, Takagahara-san. And with that, we'll move right on to the Q&A. But first, I'd like to launch a survey, which we invite everybody to fill out. We do read all your comments and appreciate any feedback you can provide. And for the Q&A, as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A tab. All right, so the first question here is, body weight can decrease even when a compound exerts toxic effects. How can we ensure that we are selecting compounds that induce weight loss through pharmacological mechanisms rather than toxicity?
Oh, okay. That's a very important point. Body weight loss can be caused by toxicity, so relying on weight alone is not sufficient. What we emphasize is confirming mechanism-based response alongside weight reduction. For instance, we use C4 staining for central acting compounds and Oximax analysis for energy expenditure mechanisms. This approach allows us to distinguish true pharmacological effects from toxicity. We have broad experience across multiple anti-obesity targets, so we would be happy to support your project. Thank you.
Fantastic. Well, thank you. All right, the next question is a good one. How should the Akshi mouse and SHC rat be used or positioned in practice?
Thank you for your question. First of all, both models develop CKD through different mechanisms, but their phenotypes are very similar to human CKD. As recommended in guidance, it is preferable to evaluate drug effects in multiple animal models. So therefore, we recommend using both Akshi and SHC, if possible. In both models, 2 to 3 weeks of administration of irbesartan improves albuminuria. So you can first run a short-term study to confirm the effect and then move to a larger study. In terms of characteristics, Akshi mice carry a genetic mutation derived from Alport syndrome patients and mainly show glomerular basement membrane abnormalities. In contrast, SHC rats develop progressive glomerulosclerosis, which is similar to FSGS. So Akshi is suitable for Alport syndrome related targets, and while SHC is suitable for FSGS related targets.
Fantastic, great answer. Okay, seeing as how time-consuming obesity studies can be, are there any strategies to shorten the evaluation timeline?
Thank you. That's an important point. DIO model typically require over 20 weeks to establish obesity. To shorten timeline, we use MC4R knockout mice, which develop obesity more rapidly due to hyperphagia. They also show less hyperglycemia than DIO models, allow more focused assessment of anti-obesity effects. This makes them a useful option when faster evaluation is needed. Thank you.
Fantastic. Thanks very much. Okay, before conducting a long-term efficacy study, I would like to know whether Axlead has gene expression patterns of our interests. Do you have any such types of data?
Yes. For the Aximers, we have generated spatial transcriptomics analysis data at 18 weeks of age, including comparison with wild-type mice. In addition, we are currently performing single nuclear RNA-seq analysis at 26 weeks of age, so we can provide information on the expression of your targets before starting an efficacy studies. For SHC rats, we have not yet confirmed or conducted comprehensive analysis. We have not conducted this analysis, but we are now currently planning to do so. Thank you.
Fantastic. All right, here's a good question here. How should we interpret and differentiate the use of emesis assays? In other words, how should we choose between them depending on our purpose?
Thank you for your question. This is an important point. Each assay differs in sensitivity, robustness, throughput, and translatability. Condition-tasty aversion is highly sensitive and reproducible, but may overestimating risk and lacks direct translational relevance. Pica behavior is commonly used but show limited robustness in our hands. On the other hand, salivary endpoints demonstrate clear response at clinically relevant dose and have better translatability. So practically strategy is to use CTA for early screening and then confirm finding using salivary-based assays. Thank you.
Perfect, great points. All right, I think we'll make this next question the last one. How is GFR measured in these models?
Yes, we generally measure GFR using FITC sinistrin clearance. The tracer is administered intravenously, and GFR is calculated based on its blood concentration over time. In addition, we also use a less invasive method that monitors tracer disappearance by using a small device without repeated blood sampling. We have confirmed a good correlation between the GFR values by these two models. And we plan to introduce this device very soon. Thank you.
Fantastic. Well, Takahara-san and Hidaka-san, thanks so much for all your insights today. It's really been a pleasure having you with us. I'd also like to give a big thanks to the audience for participating. Just a quick reminder that the recording of today's webinar will be available soon, so keep an eye out for an email giving you access to it in the near future. If you haven't already, please take a moment to complete the survey, which should appear on your screen. Again, we do read all your comments and appreciate your input. And if you still have a question, it's not too late. You can submit it on the survey, and we will pass it along to the speakers following the event. And in closing, we hope you enjoyed this Scientist.com and Axlead Drug Discovery Partners, Inc. webinar, and we'll see you again next time. Have a great day, everyone.